Hello! In this video, we will be covering weeks 3 through 8. This is the embryonic period. During the embryonic period, or the period of organogenesis, the endoderm, ectoderm, and mesoderm germ layers give rise to all tissues and organs in the body. By the end of the embryonic period, the embryo exhibits recognizable major human features, including little arms and legs and a head and face. This is also the time when the majority of birth defects occur because it's a critical phase of rapid cell division, differentiation, and organ formation. So with that being said, we will look at our germ disc from the posterior, well, not posterior, but the ectoderm view, the dorsal view. Here's that primitive node and the primitive streak. It will elongate the embryonic disc, and the notochord will elongate, and as the disc elongates, the primitive streak and the primitive node regress. Around the 3rd and 4th weeks of gestation, the notochord signals to the overlying ectoderm to thicken and form what is called the neural plate. This is also called the neuroectoderm. That's a mouthful. Neuroectoderm. And the formation of the neural plate, this marks the beginning of a process called neurulation. Essentially, what neurulation is referring to is when the neural plate forms a neural tube. So how do we go from a flat plate to a neural tube? Well, first of all, looking at our side view again of our trilaminar disc, the neural plate will form a groove. There will be some crests that will form, and with those crests will form a groove, and that'll occur along the length of the embryo. And as the edges of those crests or those folds come together, they will then form the neural tube. So if you can just imagine, kind of like folding a burrito, you've got the tortilla, the edges of the burrito coming together. Here I've got the ectoderm drawn here. You got those edges of the burrito that are coming together. There's our tortilla. And those edges are coming together, kind of like a zipper. And it'll move from a craniocaudal fashion. And as the fusion of this crest or of these folds move towards the craniocaudal end, we have these openings in our embryonic burrito, and they have names. Near the cranial end, it is called the cranial neuropore. Neuropore. And on this end, it is the caudal neuropore. The cranial neuropore will close day 25, and the caudal neuropore will close around day 28. The failure of these to close results in neural tube defects, such as anencephaly and spina bifida. And the interesting thing is many neural tube defects can be prevented if women take multivitamins containing folic acid daily during pregnancy, because folate is needed for this neural tube to close completely and these neuropores to close completely. So we now have the formation of the neurotube, and this is going to form the brain and the spinal cord, the pineal body, as well as the posterior pituitary gland. Now, as these folds are forming, as the folds or crests are coming together, neural crest cells will form. And this will be occurring along the entire length of the neural tube. And after closure of the neural tube, so here's those neural crest cells. After closure of the tube, they will undergo a epithelial to mesenchymal transition. This essentially means that they become more mobile and invasive, and they will migrate into the mesodermal tissue. And they have a lot of functions. Neural crest cells are often called the fourth germ layer due to their remarkable versatility. And we'll look at some of those functions here. So neural crest cells will form the cranial and sensory ganglia nerves, melanocytes, those will form the pigment in the skin. Neural crest cells will form the craniofacial bone and cartilage, the enteric nervous system. So they perform a lot of functions. And then finally, the remaining ectoderm will form skin ectoderm. And this will result in the formation of the epidermis of the skin, nails, sweat and sebaceous glands, mammary glands, oral epithelium, external auditory canal, and it'll also form the outer lining of the tympanic membrane. So that's the fate of the neuroectoderm. Moving on to the mesoderm. Well, no, first off, I wanted to mention this. So we have our neural tube and it's closed. By the time the neural crest cells have formed and begun their migration, there will be ectodermal thickenings that will form on, well, mostly interested in these ones on the cranial side. And these ectodermal thickenings are called placodes. This is the lens placode. And this one is called the otic placode. And this is near that cranial end. And what you can imagine is the lens placode will form the lens of the eye, and the otic placode will form a pocket. It will invaginate into the ectoderm and form the structures of the ear, like that are responsible for hearing and equilibrium. So that summarizes the ectoderm. So now moving on to the development of the mesoderm, by around the 17th day, you have the mesoderm lying alongside the notochord. Well, first off, what I want to talk about, let's just draw this. So, drawing our ectoderm, and then the endoderm. Here is our neural tube and notochord. The mesoderm, it lies along the notochord and receives signals from it and the neural tube to divide into the paraxial mesoderm. This is the paraxial mesoderm, the intermediate mesoderm, and then the lateral plate mesoderm. The paraxial mesoderm will be lying alongside the notochord, and it will organize into segments called somites. And they'll be the somites will be formed in pairs along either side of the notochord. And the first pair that will form on about day 20 to 21, and it will continue to form in a cranial to caudal sequence until all 42 to 44 pairs are formed. And this they all form by the end of week 4. So these are somites. And this is the paraxial mesoderm that is dividing into somites. The somites are a solid ball of paraxial mesoderm, and then it will form a hollow donut shape. And then from there will be some tissue differentiation. It will form the sclerotome. I don't know if I spelled that right, sclerotome. And the myotome. And finally, the dermatome. So let me just illustrate that here. So the paraxial mesoderm will differentiate into the sclerotome, the myotome, and the dermatome. And the sclerotome, draw that on either side, it will migrate to surround the neural tube and will eventually form the vertebra and the ribs. And the dermatome and the myotome, they will eventually travel to other parts of the body, say like the limbs, and the myotome will form skeletal muscle and the dermatome will form the dermis. And as these cells are migrating, a spinal nerve will also travel with them. And that's where we get dermatomes and myotomes in the adult body. This is that embryological origin of dermatomes and myotomes. So let's summarize paraxial mesoderm. So sclerotome, vertebra and ribs, myotome, skeletal muscles, and the dermatome will form the dermis and the back. The intermediate mesoderm will form the kidneys and the gonads, which are the testes and ovaries, as well as the adrenal cortex. And then finally, we have the lateral plate mesoderm, which will actually split here into 2 layers. This is the lateral plate mesoderm. This will be the parietal layer of the lateral plate mesoderm, and then this will be the visceral layer. Now, at this point, it'll be helpful to go over folding of the embryonic disc. The embryonic disc will undergo lateral folding, and it'll look something like that. But I have some illustrations of that, so I don't have to do that in real time. That'd be a nightmare. So, lateral folding will occur. In blue is the endoderm, notochord, and then here is that endoderm, and then this space here is the yolk sac. This is the amniotic cavity. And what will occur is the edges of the embryo will start to fold laterally. And you can see here, it will form along, it will form this structure here, the section below the midgut, and then right at the, if we took a transverse section, right at the level of the yolk sac, we would have this connection to the yolk sac. But I'm going to be redrawing this to illustrate the lateral plate mesoderm. So we have our lateral folding. Here is that surface or skin ectoderm, and then the parietal lateral plate will look something like this. This will be that visceral lateral plate. Here is the neural tube. component of that notochord right there. And then, if you can see, we have the endoderm that has now formed a tube. But looking at the parietal layer and the visceral layer, the parietal layer will form the body wall. It will also form the dermis of the body wall and limbs. It will form the bones and connective tissue of the limbs as well, the sternum, and the parietal serous membranes. This could be like the parietal layer of the pericardium, of the pleura, the peritoneum. The visceral layer will form the cardiovascular system, blood cells, smooth muscle of the gastrointestinal tract and the respiratory tract, as well as the spleen and lymphatics. So that covers our discussion on the mesoderm. And finally, we'll now start talking about the endoderm. And with the lateral folding, we have this tube structure here. The endoderm has now formed a tube. And then there's also a connection at the mid portion of that tube that is connecting to the yolk sac. And its connection is through a structure called the vitelline duct. Yes, the vitelline. Oh, watching the spelling. Vitelline duct. And the yolk sac, it's a transitory structure. It contributes some of the first blood cells. It also houses germ cells. But that's looking at from a sagittal perspective. So not only will the embryo undergo lateral folding, it'll undergo craniocaudal folding. So this is the cranial end of the embryo. This is the caudal end or tail end of the embryo. I would highly recommend finding some animations to help clear up what this craniocaudal and lateral folding look like. But if this makes sense, that's awesome. This is the amniotic cavity. Here's our yolk sac. This is the allantois that is extending into the connecting stalk, which is our future umbilical cord. So the embryo will kind of curl forward. Its head and tail will curl forward, kind of forming that C shape. And as it is curling forward, the allantois, it will be coming closer and closer to this region of the gut tube. Let me go over the regions of the gut tube real quick. So here is our endoderm gut tube. This is the foregut, the midgut, and then the hindgut. And the allantois is coming off of the hindgut. And as we have this craniocaudal folding, the allantois will be coming closer and closer to the hindgut and the main body of the embryo, and it'll eventually become incorporated into the body and form what is called the cloaca. It will also be partially fused with the connecting stalk here. I hope that wasn't thoroughly confusing. One last thing I want to touch upon, at the ends of the embryo, the cranial and caudal ends, there are ectodermal, endodermal membranes. The cranial end of this membrane is called the oropharyngeal membrane, and it'll rupture to form the opening of the future mouth. On this end, the caudal end, it is called the cloacal membrane, and that will break down on the 7th week to form the anal canal. So let's just summarize now what the endoderm is going to form. So I kind of already gave it away, but it'll form the epithelium of the GI tract as well as the epithelium of the respiratory tract. It'll form the urinary bladder, the functional tissue of the thyroid, parathyroids, and the liver. It'll form the pancreas, the tonsils, as well as the lining of the tympanic cavity and auditory tube. So here is just a summary of what each of those layers of the trilaminar disc are going to form in weeks 3 through 8. And just a reminder that weeks 3 through 8, this embryonic period is a critical period where all tissues and organs in the human body are going to form. Enjoy studying, and I hope this video at least somewhat helped.